Okay, so this is the third video on cell signaling, and it's about G-protein coupled receptors. I think G-protein coupled receptors are really, really cool. So I really suggest that you watch the G-protein coupled receptors video, actually a couple of videos, and we're going to talk about the general principles of G-protein coupled receptors, and then maybe do a couple of specifics. Okay, so G-protein coupled receptors are the 7-pass transmembrane proteins. On the extracellular side, they have the ligand binding domain, and on the intracellular side, they are able to bind to the G-alpha subunit. And then the G-alpha subunit is then bound to a beta-gamma subunit. So there's three proteins: the alpha, the beta, and the gamma make up the G protein, and then the G protein coupled receptor is a fourth gene product. Very often, the alpha is linked to the membrane, and then the gamma is linked to the membrane, and the beta is always associated with the gamma. So it's always alpha, beta, gamma, and receptor. So sometimes the G protein is already associated with the receptor, and when the ligand binds to the receptor, that causes a change in shape that causes the G protein to dissociate from the receptor and go do its work. Other times, it's not bound already, and the ligand binding to the G protein coupled receptor recruits the G protein to the receptor. So there's lots of different ways that this could go. Don't get too rigid in your thinking about G protein coupled receptors. But in every case, what we're going to have is the signaling molecule causing a change in shape of the G protein coupled receptor, and that change in shape is going to activate the G protein. And when the G protein becomes activated, it was previously GDP bound, but the change in shape of the receptor causes a change in shape of the G alpha subunit, and the GDP is exchanged for GTP. Now the GTP bound active alpha subunit is now activated and can go on to maybe activate an enzyme or do some downstream work. The beta gamma dissociates from the alpha when the alpha is activated, and maybe the beta gamma can go do some work, maybe cause an enzyme to become activated. So the take-home message here is that the G protein coupled receptor is called a G protein coupled receptor because it's bound to GDP or GTP. So when it becomes activated, it exchanges its GDP for GTP, and that activation allows for the activated alpha or the activated beta gamma or both to go on to do something else. So what could that something else be? Well, let's take, for instance, another enzyme. So you got some kind of enzyme that the activated G alpha subunit may go and turn on, and then while the G alpha subunit is bound to it, this enzyme is on. So the GTP bound has a conformational change that allows it to activate or bind to and activate an enzyme, and then that enzyme causes some sort of intracellular signaling cascade. Then over time, the G alpha subunit may hydrolyze its GTP to GDP, and that's going to cause it to go back to the inactive form. So again, watch this video. I'll fix the link, but watch this video, and they go through several different iterations. One of those iterations is maybe something like that the beta gamma is the one that becomes that does the downstream signaling. So it's still the same here, that when the G protein coupled receptor binds to its ligand and goes through its conformational change, it causes the G alpha subunit to exchange its GDP for GTP. But in this case, the activated alpha subunit isn't the one that's doing something downstream, but instead the activated beta gamma does something. So when the alpha subunit binds to GTP, it will dissociate from the beta gamma. Now the beta gamma is free to go and do something else, like maybe in this case open a potassium channel. And so we know that opening a potassium channel is going to cause a change in membrane voltage, and so there will be a physiologic effect downstream of the beta gamma binding to it. Again, the G alpha subunit is going to hydrolyze its GTP to GDP, and once it's GDP bound, then it goes through that conformational change to go back to the form that binds to the beta gamma, and when beta gamma is bound to the alpha, it is no longer active and so stops gating open the potassium channel. This is exactly how that one that I told you in the heart, this is exactly how that works. This is the metabotropic, it's called a muscarinic acetylcholine receptor. It's a metabotropic acetylcholine receptor in the heart, and when acetylcholine binds to the muscarinic receptor, the G alpha subunit exchanges its GDP for GTP, and that releases the beta gamma to go open the potassium channel, and that change in membrane voltage is going to cause the heart to slow down. This is that Lowy experiment with the two frog hearts that was the first demonstration of neurotransmitters. The stuff that he called vagus stuff was the acetylcholine released by the vagus nerve that slowed down the heart. So we talked about that before, and now we know a little bit more about the actual mechanism by which the acetylcholine was causing the heart to slow down. And we did talk about that before. The first proof of the existence of neurotransmitters and Lowy's Nobel Prize. So it doesn't have to be the beta gamma opening up a potassium channel. We talked a second ago about the G alpha subunit, when activated by being bound to a GTP, can cause an enzyme to be activated, maybe changing an inactive form to an active form, or some sort of intracellular messenger system. An example of that is the cyclic AMP system. So when the G alpha subunit is bound to GTP, it activates an enzyme called adenylene cyclase. And adenylene cyclase is going to take ATP and turn it into cyclic AMP. And cyclic AMP is going to bind to the inhibitors of pKa. When so two pKa's and two inhibitors are in an inactive form, cyclic AMP binds to the inhibitors and causes conformational change, and then the activated pKa is now free to go and do downstream work. Since pKa stands for protein kinase A, well, we know what the downstream work it's going to be doing is it's going to be phosphorylating stuff. So it could phosphorylate the phosphorylase kinase in this example, and phosphorylase kinase is going to be able to then phosphorylate other things like the inactive glycogen phosphorylase. So here, adrenaline is causing a cyclic AMP cascade, 2 amplification steps, the downstream effect of which is glycogen breakdown. And of course, glycogen breakdown is going to cause there to be more glucose in your bloodstream. In response to adrenaline, we would want more energy for our muscles to be able to support that fight or flight. Another thing that cyclic AMP might be doing to the inactive pKa, or the activating the pKa, is causing pKa to go into the nucleus, where it is going to phosphorylate a transcription factor, maybe like Krebs or something like that. And so the inactive transcription factor, once phosphorylated, becomes an active transcription factor and causes transcription of some gene product. So in one cell, we might see the more glycogen breakdown and more glucose, but the exact same signal, adrenaline, through the exact same pathway, cyclic AMP, might in another cell cause some gene to be transcribed, like a stress hormone or maybe cytoskeletal element or something like that. So watch this second video. Again, I'm going to have to fix the link. But watch the second video about pKa. Okay. Another G protein couple receptor is the PIP pathway. So inositol phospholipid pathway. So once the activated G protein goes and activates its downstream target, in this case, the activated downstream target is phospholipase C. And phospholipase C is going to go to an inositol phospholipid and cut it, so that now we have IP3, inositol 1,4,5 triphosphate, and a diacylglycerol. So those two downstream signaling molecules are going to do 2 different things. First, the IP3 is going to gate open calcium channels on the endoplasmic reticulum. And so that's going to cause intracellular calcium, cytosolic calcium, to go up. And this wave of calcium is going to then find an activated PKC. Well, how did PKC become activated? The diacylglycerol that was cut off of the inositol phospholipid is going to, once there's a diacylglycerol, it's going to be able to activate the PKC. But PKC doesn't activate from diacylglycerol alone. It also needs to bind to calcium. So both of those, diacylglycerol and the calcium that is downstream of the IP3, are required to activate the PKC. So we see these calcium signaling in many different biological processes, and so I think you should watch the video on calcium also. So this is G protein couple receptors. There are several different versions of G protein couple receptors that I'd like you to know for the exam, as well as how do we turn them off, because that's going to be really important for when we can't turn them off later in things like cancer. Okay, so that's video 3. You can go ahead and move on to the video 4, the receptor tyrosine kinases video.